Greetings and humble salutations. My name is Raven Hollis, and I'm a recent graduate from Prairie View A&M University, and I did research at the Columbia University Irving Medical Center under Dr. Schvartzman and Grace Forsyth, a lab manager. I graduated in May of 2022, and my topic is Sensors of Metabolic Stress, Transcription Factors in a Harsh Environment. I was at Columbia as an Ernest E. Just biomedical scholar. What is hypoxia? Now, before we get started, take a deep breath in and out. A lot of people don't understand the role oxygen plays in our environment. Oxygen actually makes up 21% of our atmosphere. However, in our body, this varies. How can we define hypoxia? Hypoxia can be defined as a deficient amount of oxygen reaching tissues to maintain equilibrium. When your body is not at equilibrium, it will do anything it can to make sure it gets back to that state. Now let's get into the purpose of my research. We see that it says HIF1 alpha's role in cancer and hypoxia plays a role in patient outcomes. In relation to cancer, hypoxia is a common characteristic of solid tumors that induces changes to gene expression, metabolism, and therapeutic response. Now, you might be wondering, what is HIF1 alpha? HIF1 alpha stands for Hypoxia Inducible Factor 1 alpha. One major driver of the changes we discuss are the inhibition of degradation of the transcription factor HIF1 alpha. The study aims to help us understand how HIF1 alpha is able to assess its targets within repressive chromatin environments associated with hypoxic conditions. On a broad scale, this research is applicable to outcomes in patients, as stated on the slide. Now, let's get into our design methods. We have CRISPR-Cas9 and Western blotting. We generated a CRISPR-Cas9 knockout of HIF1 alpha and renal clear cell carcinoma cell lines. When using CRISPR-Cas9, we use it when we to target and look for specific DNA. When the target DNA is found, Cas9, one of the enzymes produced by the CRISPR system, binds to the DNA and cuts it, shutting the targeted gene off. Then we're able to validate the knockout of HIF1 alpha by Western blotting. Western blotting is a laboratory technique used to identify and separate proteins. And as you can see, we have two different images to depict CRISPR-Cas9 and Western blotting. Now let's get into the nitty-gritty, our results and conclusion. Our results. This is still an ongoing process, so results are still pending. However, we're able to create a new cell line and create clones for the guides. When we get into discussion, there are a few questions that we want to answer, but before that, our future directions of this project entail utilizing cut and run. This is actually a technique that will allow us to identify the sequences of DNA with which HIF1 alpha is interacting. And the questions we want to answer are, can HIF activate its targets and regions of heterochromatin? And do certain chromatin modifying enzymes specifically aid in HIF in activating its targets? These are some of the questions we want to answer in the future for this project. I'd like to go into acknowledgements. Dr. Schvartzman, my PI, and Grace Forsyth, lab manager, and the UNCF Foundation as an E. Just biomedical scholar. Thank you so much.